Hello, everyone. This is the review recording for exam 2, and it'll probably come in sections just based on the amount of time that I have to do this. But we might as well get a good start on it now. So once again, I just lecture directly from the exam, and you'll have an idea what's going on. And the first topic, something I told you to expect, here's the answer key: simple diffusion, facilitated diffusion, active transport, osmosis, phagocytosis. So again, it's basically a modified matching, used once, twice, or not at all. I'm going to describe the activity, whether it's solvent or solute moving, whether it's water soluble or fat soluble, whether it's going through a channel, whether the channel opens, whether energy is required, distinctions between phagocytosis and pinocytosis, and receptor-mediated endocytosis and exocytosis, exactly what all of those are. Those are some of the topics that you would see there. So there's in each one of these little phrases or sentences, you have to pick out what it describes. So you have to understand. So immediately understand, distinguish. If it says it's an active process, it involves energy expenditure, and so it has to be active transport. If it's involving endo or exocytosis, again, you're opening and closing membrane, active transport. Everything else pretty much is passive. Solvent implies water, so you would probably see osmosis. So that takes care of that. There's several questions regarding the cell cycle and the different phases. So you should got to know what happens in each phase. So start with the interphase, G1, S, and G2. G1, I can't do the little subscription, so I just put G with a 1 next to a G with a 0, etc. So effectively what occurs, G1 is the big one where you're preparing for everything. You're building more nucleotides, you're building more membrane, you're building more proteins, and everything else that you're building, you need to make all those structures. Okay, S phase is effectively only making the duplicate strand of DNA that's there. So that's obviously where mutations occur, because mutations are error in DNA. That's where you would make them. And G2 is final preparations. So remember what occurs in all those events. And obviously, this dovetails into what's going on in M phase. The quote mitotic phase involves not only mitosis, and again, nuclear division, as well as cytokinesis, cellular division, even though the processes overlap slightly, mostly in anaphase. So I use PMAT, prophase, metaphase, anaphase, telophase, and the big deal here is what are the 4 events I underscore that happen in prophase, in no specific order. The chromosomes supercoil, which means they appear. The nuclear membrane disappears. The nucleolus disappears. And lastly, that spindle starts to form. Those are all reversed in telophase. Metaphase in the middle means they line up in the midline. Easiest phase to recognize. Anaphase is the shortest, they separate. Also, anaphase is where we sometimes begin to see the beginning of cytokinesis, that cleft or cleavage furrow, as effectively tiny fibers inside the cells are starting to basically create and sort of pinch off and separate what will eventually be the 2 cell membranes. And effectively when that process completely concludes, we have the end of the cell cycle, we start all over again. So those are the big things. So again, one of the matchings would have prophase, metaphase, telophase, anaphase, cleavage. Okay. You can say cytokinesis just as easily as cleavage, I suppose. And again, I'll have a phrase or a description or something like that. And in that point, you need to pick the appropriate answer. The difference in tonicity is important. So we have 2 different ways to sort of identify degrees of tonicity. Okay. I mean, effectively what they do would be one way, but really the description, it's the relationship of solute to solvent. So when something is isotonic, same tonicity, it means it has the same concentration or the same relationship between solutes, all, to solvent, water, in our bodies. And effectively, it's no different than the dog who's sleeping on the couch next to me, or the cat who's prowling around and probably sleeping upstairs, or the other cat doing the same thing. So all members of the animal kingdom have this, and basically that relationship means that water doesn't move effectively, forcing its way and exploding a cell, okay, or being sucked out of a cell and having it shrink, a process we call crenation. Basically, so anything we add to that, it may happen transiently, we may get some little things, but basically the idea is so solutions inside the cell, oh, they have a different composition than outside the cell or in the tissues or in the bloodstream. They have different compositions, but they all have the same relationship of solutes to solvent. That's the whole idea of what isotonic is. Hypertonic means there's more stuff. Think salt water compared to lightly salted water, or completely plain water would be hypotonic. It really has very, very little dissolved in it. And obviously, that's going to push water in. And something like seawater is going to suck water out and cause cells to crenate. Also, along with that idea, and I forgot to mention it in the process of the word apoptosis. Apoptosis is programmed cell death. Is what happens when cells should basically, let's say there's a bunch of mutations in the cell cycle, and all of those, and what's effectively happening, that cell is never going to complete those signals to go into mitosis, so it undergoes apoptosis. So it's a safeguard for cells that are really likely to become cancerous, aberrant, or basically a cell that won't do its job and likely to cause significant problems. Obviously, biologically, the more cells you make like that, okay, if there's a fault or drugs are involved or excessive alcohol is involved, the more cells you make like that, the less the apoptosis is effective. It just can't clobber all the cells and you start getting bad ones. So all of those are important factors that are where, and the whole idea of the cell cycle is this is all about mitosis. It's all about making identical copies. Yes, the DNA is the same on both sides. One's right side up, one's upside down. So that double strand of DNA is identical. One's top to bottom, one's bottom to top. Again, the fancy name for that is anti-parallel. And when we make a new cell, we do it semi-conservatively, meaning we conserve half of the DNA. So the new cell has half the original DNA and half new DNA, and the new cell has half of that original DNA and new DNA. I may have misspoken that, but effectively the two new cells have one strand of original DNA and the other strand of now newly synthesized or made DNA. That effectively keeps the DNA, relatively speaking, fresh. Okay. So we're not making continuously new stuff because eventually, effectively, it undergoes a process of degradation. So that's something to remember. Where when we talk about meiosis, typically in repro physiology, the whole idea is diversity. We don't want to make the same DNA in those cells. We want to make something that's new. We're going to have some similarities, but there's going to be new. But that's not part of this exam. So meiosis isn't, mitosis is. That's what this is all about. And I went into a lot about cancer extensively. I think to me, it's obviously it's very important to me because I've had the diagnosis, but it's important to everybody. It's just as you get older, your level of fear increases dramatically when the word cancer is involved because you're getting closer to when it could happen. And what happens? The cell cycle loses its controls. There's too many bad cells, so apoptosis can't occur or is unable to occur. There's a number, the mutations are very significant. They're not isolated, what we call silent mutations. Okay, the cell begins to grow aberrantly if that occurs. It invades healthy tissue. It doesn't appreciate those boundaries. Like what's we might remember us discussing contact inhibition. When the cell is kind of cuddled together with all the other cells, normally it stays there. But a cancer cell will just grow out of control, gobble up the other ones. It'll heap up. It's really extraordinarily. And remember, a lot of those controls are based on those things called cyclins. They're proteins that produce almost as a waste product at the end of G1 and at the end of G2. They bond with something called cyclin-dependent kinases, or CDKs. Those serve to unlock those stops, those restriction points at the cell cycle at G1 and at G2, and allow the cell cycle to proceed. Once it undergoes mitosis, all of those products are destroyed, and we basically start all over again. So it's not like if you have abundance of them, they're going to help the next cycle. It doesn't work that way. So they're strictly associated with cyclins are strictly associated with rather the cell cycle, and specifically we're talking about the mitosis or mitotic cell cycle, to use the appropriate term. So that's all part of it. And then we spent a lot of time, and I'm not going crazy with the questions about it in this, with regard to the polarity of the cell. Okay. Because really, in the next chapter, when we start to do muscles, which we're going to start, if I'm dictating this Sunday afternoon, the 18th, rather than the 19th, and on the 20th, we're going to start muscles, which is the next unit, the third of our 4 units. And effectively, it all based on the idea that cells all have polarity, meaning just across the membrane, the interior of the cell is neutral. The exterior of the other cells or the interstitial fluid, all of those things effectively are neutral. But right along the cell membrane, because of those potassium and sodium leak channels, and again, potassium is the big player, okay, because there's more of those leak channels. Effectively, what happens is that we create a slightly more positive environment on the outside and a slightly less positive or more negative environment on the inside, directly on the membrane, directly outside of it and directly inside of it. So if we would say across the membrane, and it works out to something about a minus 70 millivolts. That's because of the ability for potassium to leak out and for sodium to leak back in. More potassium leaks out, and then in an attempt to compensate for that, a certain amount of sodium leaks in. It gets down to that minus 70 millivolts. And so they're all going through effectively a kind of integral protein that is a leak channel. Okay. It's part of the membrane. And if you go back and look at those animations, they're just all over the place. So continuously in all cells, and particularly in excitable cells, because that is always changing rapidly, we have those sodium-potassium pumps. And what the pump is doing in every cell is basically making sure, we lost all that. I got to pull the sodium back in. That's active transport. I've got to kick sodiums back out. That's active transport. So we call them pumps. The name pump implies active transport. Pumps require energy input that are there. And I think that's a really important point. So not only in excitable, they're actually found in all cells. The key issue, again, potassium is the key player in this. And polarity just means charge across the membrane. That's all. And that's in the context of what we're describing in this. It's not the same as the in an epithelial cell, which you do in an anatomy lab. You have the basal portion and the apical portion. Even though that's kind of another term we often use polarity, it does not apply for what we're doing here in lecture. Do not be confused about that. So I think that's all important to recall. And so those are, so for instance, in that first roughly 20 plus questions, it's all about the cell. So I'm trying to give you an idea. Maybe the next, I'm going to look here, probably the next, let's say another 20 questions or so. Okay. Maybe 23, 24 questions or so. Okay. You're going to basically, that's about bone. So far it's like 22, and then maybe 23, 24 about bone. And then from arthritis, I would dare say you have at least 10, maybe 12. Yeah, about that. And maybe a few more odds and ends at the end. So maybe it's, if I'm thinking for the 60 questions, let's say it's probably fair to say maybe 25 to 30 bone, maybe okay, so let's say 25 plus about bone, maybe 22 to 25 about the cell, maybe roughly 10, maybe 12 about rather joints. Okay, that's the distribution. I think that's important for me to tell you. So it gives you an idea what to study. It's not to de-emphasize anything; they're all obviously important, but it gives you the idea. It's fairly balanced between bone and cell stuff, and maybe half as many vis-a-vis arthritis. And the arthritis, it should be fresher in your mind too, so maybe you'll be able to recall it more easily. But back to bone. So what bone does? Know all the stuff it does. Hematopoiesis, structural support, protection, calcium storage, blah, 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 blah. All of that type of stuff. Okay. So understand those are the basic things you see that in every chapter, in every PowerPoint, in every lecture. Basically, why bone is important and why is bone and bone is made from cartilage. We all know that the only cartilage it's made from is hyaline cartilage. Remember the description of that. Okay. Very flexible, resilient. It's by far and away when it comes to cartilage in the skeleton, it's number one on the hip rate. You have very, very limited elastic cartilage, and the fibrocartilage stuff is very limited too, like intervertebral disc, symphysis pubis, that kind of stuff. Knee discs, etc. Okay. And so, even though most of our studies are about long bone, the flat bone, which is important, remember that's like a sandwich, cortical on the outside, cancellous on the inside. It's important to know the distinction because everything else are effectively long bones. Yes, there's some oddball ones. The short bones have a lot of cartilage, but they're kind of little cubic, more or less structures with cartilage all over the place. They're an oddball. And it's the same with the irregular bones, like the vertebrae. It's sort of a hybrid. It's an oddball. But the long bone is when we think about the epiphyseal plate. The long bone is when we think about that remarkable, really, we identify cortical bone with that. And again, cortical or compact bone has those layers. They look like the tree trunks. We call them lamellae, which means layers. Anyway, lamellar bone would be another term. And then in the interior, you have the cancellous bone, which has those interesting plates, that trabecular, that honeycomb-like pattern that you saw so distinctly on many of the illustrations. And I took great pains. So I really spent a lot of time trying to do the descriptions for you. And really, my underscore with a lot of this is what's around all bones, but particularly the shaft of long bones, is called periosteum. Periosteum is just it. Okay. Nearly everything that makes more bone, your ability to make and break down bone comes from the presence of what they call osteoprogenitor cells. Effectively, they're osteoblasts along that in the periosteum on the lining as it's facing the bone. You have osteoclasts, those groups of cells with the ruffled borders. There are really modified white blood cells that gobble up the bone. And it's just a continuous process. We make more, we take away more. We make more, we take away more based on the usage of the bone, the pressures that are on it. And it's basically a continuing process of remodeling that goes on. And again, so that's really, to me, the really important structure, and then particularly understand the osteon, the main structural unit of bone. That plays a pivotal role because it's it's really those little pillars, and those little pillars are basically effectively osteons. And they have that central canal, and then they have those layers of bone, and in those layers of bone, which are formed with collagen fibers spiraling in one direction and then the other direction, those calcium binding proteins that makes up the ground substance, the fibers, the matrix, and then it absorbs those mostly calcium phosphate salts. Remember the fancy name hydroxyapatite because you're going to see it again. And what they do is that gives bone its remarkably rigid structure. Yet, because of the nature of the way that since it's those individual pillars, and then you have interesting thing called interstitial lamellae. They're like little bits of glue, taking those basically somewhat flexible cylinders and holding them together inside, and they're surrounded by circumferential layers or circumferential lamellae, and they kind of hold it all together. So, just like I tried to show you in class, you might take a whole handful of pencils, put some glue on them, and it'll hold. But then now you put some rubber bands around it and it'll hold even better. That's kind of what cortical bone is. And so each of those have that very, very interesting structure. And I love that idea about the collagen fibers going clockwise and counterclockwise and clockwise and counterclockwise. So they're tree-like concentric rings we call lamellae. Remember, all of this is forming from a cartilage cell. That's what we call it enchondral or endochondral bone. It means bone is formed from effectively a model that's made of hyaline cartilage. That's all. That's all. And when it's growing, we see a growth plate, a clear zone that separates the shaft from the ends. The shaft is the diaphysis, the ends are the epiphyses. And we spent a fair amount of time talking about those three hormones that play a role. Okay. The biggest player of all is parathyroid hormone by far. That's the one that's active in breaking down the bone, okay, increasing the calcium in the bloodstream. It does so by activating those osteoclasts. They secrete those chemicals. They gobble up the bone, not just leach out the calcium, but they gobble up the bone substance and release it. And again, remodeling takes place, but effectively, this is the way we're constantly adjusting the calcium levels. When we need more calcium, we suck it out of bone with those osteoclasts. When we don't, when we have no greater need for it, or the need begins to lessen, we start putting it back in the calcium. In children, that calcitonin, while the bone is growing, probably plays a good sized role. Other than that, probably in adults, it just happens naturally as we begin to reform bone. I think that might be the best way to express it, since we don't really know the role of calcitonin. Remember, calcitonin comes from the thyroid, from a special cell in the thyroid. It's not the thyroid's major function, where the parathyroid gland just makes parathyroid hormone. It works on three areas: on the bone directly, as I just explained, on the kidneys, basically telling the kidneys to hold on to calcium, don't excrete it in the urine, and most importantly, of the ones that other than the bone function, is how do we get calcium in our systems anyway? Through our diet. And in order to get in our diet, we got to have vitamin D. So that's the third hormone: parathyroid hormone, vitamin D, and calcitonin. Vitamin D has to be activated by sunlight. That's why it plays such a big role in osteoporosis. So all of that's really relevant. That's there. And as long as we have active vitamin D, we can absorb the calcium with what we eat, okay, or drink, or whatever, wherever the calcium is located. And certainly, our culture are abundant in dairy products. Those are all very good sources of calcium, but you got to have the vitamin D to go with it. So that's really important. I always go over the top about fractures, and an interruption in the overall integrity of the bone. Anything like that is a fracture, without going crazy. Look at fracture healing as well. Remember that initial first phase, maybe it's a day, maybe it's two, maybe it's three, where it forms that hematoma. Okay, it hurts, it's painful, but that hematoma, as it begins to start to solidify and turn into a gel, that really sets the framework for that, eventually, that callus formation. And the callus formation is initially, it's just like bone is made. It starts kind of fibrous, then it becomes cartilaginous, and eventually you go from that fibrocartilaginous callus formation into a bony callus formation, and then the bone heals in a process we call union. And remember the different kinds of fractures: open and closed, open exposed to the air. Sometimes the bone is actually protruding. Those are very nasty. And those, you have to treat them with antibiotics because they're going to get infected, or they're actually presumed to be infected, would be a better way to describe it. And then closed fractures, the beauty of that is that even they may be very, very difficult to treat, unless you can treat them aggressively right away, because you're not in the back of your mind going, I better make sure we don't have an infection before I start to reconstruct or fix the fracture. Fancy term we call that. We call them reductions. Reduce the deformity of the fracture, and you're doing a reduction. So then you have all those classifications: is it complete or incomplete? In other words, does it go all the way through or not? And incomplete fractures in children we call green stick fractures. And you remember the analogy from class, like if you try to snap the piece of wood and it's living wood or new wood, it's kind of green inside and it bends rather than snaps. And old wood snaps, and sort of, you're looking for firewood to make a fire, a campfire, or a fire pit outside your home. Okay? So all of that becomes really important. And then different definitions. Is it displaced? Is it angulated? Okay. Those are also part of it. They give you an idea of how displaced means the fragments are pulled apart. Are they in a line, or are they angled at a cockeyed angle? That would be angulated. Remember, if there's one fracture line, there's two fragments. If there's two fracture lines, you have three or more fragments. That is called comminuted. Do not confuse that for compound. Compound is another name for open. They are synonymous. So you can have, in a really bad fracture that's where the bone's sticking out, you could have a compound comminuted fracture. Multiple fracture lines and oh my goodness, bone exposed to the air. So that's when it comes to fractures, that's kind of the five-alarm fire of fractures, really bad. So those are, again, other things that can occur. So I think you need to understand all of those parts of things vis-a-vis fracture, the different terminology, growth plate fractures, and where the typical area that's weakest, that zone where the cartilage is calcifying. Why fractures they heal so well is because the magnificent bone supply. I think those are all really important factors. Again, bone has a remarkable relationship. It's about 2 to 1. It's about 2/3 organic salts. I mean, I beg your pardon, inorganic salts, and about 1/3 of the osteoid, the organic material. So and that's based on weight. So bone's heavy because of all those calcium phosphate molecules and other things, other calcium salts or some magnesium salts that are in there. That gives bone its strength. And bone's probably not as heavy as you think. Again, I think muscle is much more about our weight than bone is about our weight. Bone's kind of interesting in that regard. So I think you have to remember all of that. And again, don't forget the definition of osteoporosis. It's a loss of it's not just calcium not being there or calcium salts. It's a loss of bone substance, both the organic and the inorganic. Is it highlighted by the inorganic? Sure, we see that radiographically, but all of the bone is being lost. It's just that we were able to visualize the radiographically or with scans that loss of the inorganic material that's there. So I think that's important as well. Let's see what else. All the risk factors for osteoporosis that are there. Menopause certainly is number one in the hit parade. Having a sufficient amount of exposure to sunlight. Uh, okay, if you don't have a lot of sunlight, that's a risk factor. If you're inactive, that's a risk factor. If you don't have a sufficient amount of dietary calcium, that's a risk factor. All of those kinds of things. Okay, if you don't have enough vitamin D, that's a risk factor. All of those are sort of part of the process for this. I guess that's the best way I can describe it to you. What's going on? Let me see if I can find other stuff about bone that would be important. Here, healing. Just talking to myself. It's okay. And then the arthritic things. Again, arthritis, very, very common. One of the most common diagnoses you'll ever see. The most common type by far is osteoarthritis. That's the wear and tear arthritis most of us get. True inflammatory arthritis is caused by your immune system and is typically an autoimmune phenomena. There's some other oddballs, but it's really an autoimmune phenomena that is causing effectively your white blood cells or their byproducts to attack the joints. In autoimmune diseases in general, the body attacks itself. In this case, it's highlighted by attacks that are going on in the joints. And there's a lot of variations, and you may learn about it in the immune system chapter. We talk a little bit about it in micro, that kind of stuff. But the first sign, and the difference between the two, the first sign, because the white blood cells, is they cause swelling in the joint and effectively lead to instability in the joint. They cause the joint to look washed out, really losing its calcium salts. Okay. And they're all very painful. They're all inflamed by nature. The osteoarthritis is the same thing. It's inflamed and painful, but it's not because of the immune system. It's mostly wear and tear. It's sometimes occupational. It can come from accidents. It can come from sports. And the first sign is narrowing of the joint. It's not as wide from from bone to bone, so the joint space gets narrow. The ends of the joint begin to thicken. They get more radiodense. They begin to square off, would be the way we would describe it radiographically. And eventually it just doesn't work at all. So you kind of lose joint space, where the highlight and the true inflammatory, like rheumatoid arthritis, the joint becomes unstable and separates. Oh, some other stuff in bone I forgot about. Don't forget the canaliculi. Those are those little mini canals that the bone cells, remember the bone cells are all in those with lacunae, those would appear to be little spaces that surround them. You have those little, it almost looks like little hairs. Those are where those little tentacle-like projections of the osteocytes kind of touch each other, send chemical or electrical signals to one another. And the Volkmann's canals, those are the canals that go tangentially, in other words, at right angles to the central canal, which is along the long axis, but point toward the center of the bone. Those are filled with blood vessels instead. So again, the central canals are sometimes called Haversian canals, or that's the Haversian system, is the way it's tried. Some people say Haversham, and the Volkmann's canals are at right angles. Let's see, almost forgot that. Okay. And then a little bit about joints. I do have one of those things with the different types of joints. So for the ones that do not have a joint space, those are the fibrous ones and the cartilage joints. So whether it's a symphysis, whether it's a suture, whether it's a gomphosis, doesn't matter. Okay, they don't have a joint space. Again, the joint does not have to move by definition. Synovial joints, the ones we talk about with the elbow and the knee and your fingers and your hips and shoulders, those are all movable joints. Those are synovial joints. So I think that's the first thing you have to remember. And don't forget the difference with all the ligaments. Some ligaments are part of the capsule, some ligaments are inside the capsule, and some ligaments are really outside of the capsule, really than being just a thickening of it, part of it. And the knee has all three. So remember, they're intra-articular, okay, articular, and extra-articular. I mean, if you want to think of it that way, the fancy names. But the only one that has the intra-articular ligaments that I'm aware of, strike that. The intra-articular ligaments are the knee, and those are the cruciate ligaments. And the other one, I guess you could classify that way, is that long head of the biceps, that sort of super stabilizer in the shoulder. But still, that's a tendon, so I just strike that. But cruciate ligaments are intra-articular. I'm just sort of talking off the top of my head with this. Let's see what else. And when we get there, I'll talk about gout to you, and it involves crystals, and it causes a lot of inflammation from those crystals. The crystals form and settle in the joints, and they're caused by an abundance of a chemical called uric acid in your bloodstream. It can be, mostly there's a hereditary basis. Lifestyle can play a role, and I don't think it's such a big deal. Usually has to do maybe with an enzyme lack, but we'll get into that on Monday if you're listening to this tonight on Sunday, or we will have gotten into it on Monday the 20th. And don't forget, though, I forgot to mention about the joints. Large joints without a real pattern, asymmetric, means it's not necessarily the same on both sides, favor osteoarthritis, the knee, the elbow, the shoulder, the hip. Okay. Where small joints, bilateral and symmetrical, fingers and toes, wrists and ankles, that's rheumatoid arthritis. And I think that's where you'll find that as well. Let's see. Oh, yeah, and I have one. And again, I told you you're going to have one of these. So here's the key: hinged, condyloid, pivoting, gliding, sliding, saddle. And I'm going to give you a joint. You figure out which one of it is. I mean, it's all joints we mentioned and talked about. Interesting, I got done with the review. So that concludes the review. Thank you for your attention. If you have any questions, just shoot me an email or ask me in class. Bye bye.